So, as Bill mentioned, my name is Joey. I'm a first-year graduate student at Rice University in the lab of George Phillips. And so today I'll be talking about the facilitation of membrane protein serial femtosecond crystallography using this lipidic cubic phase injector. So, as you all know, membrane proteins carry out a lot of critical roles in cellular environments, and they make up 1/3 of the proteome of most living organisms. And there are studies that show that they constitute 60% of current drug targets in humans. And so it's important to obtain structures of these membrane proteins to sort of get an insight for their function, and also to sort of be able to design drugs for them. And so here are just some examples that I'm showing of different membrane proteins. And so there's a transporter, right? There's like proton pumps. Here's a receptor that has a signaling cascade going. And so this is sort of an example of the protein that was used in the study as a proof of principle. And so the protein used here is like the human smoothened receptor. And so that's involved in a signaling pathway called the sonic hedgehog pathway. But then again, here are just two more examples of, they carry out enzyme reactions and they anchor things. So membrane protein crystallization has historically been difficult, right? So traditionally it was done in detergent micelle solution up until about 20 years ago when LCP was introduced in 1996. And so before that it would involve extracting membrane proteins from their native lipid bilayer. And that's sort of like not a favorable environment for these proteins, right? They're usually in this lipid bilayer where you get this lateral pressure. And so that's one of the things the paper mentioned, that removing them from this lateral pressure can affect their stability and cause them to aggregate. And so that sort of makes sense when you think about these like helices that span a membrane, right? Like the lateral pressure might be helpful to keeping those proteins well folded. And so the idea was that using LCP, you might be able to get these proteins, I guess, more happy in a more favorable environment by providing this lateral pressure with the LCP, and also it would be a more native-like environment because it's a lipid membrane. And so on the right here, I'm just showing this plot of sort of a trend expected for the number of structures that we would have after year 0 of the first soluble protein and the first membrane protein. And so you can see that membrane proteins lag behind soluble proteins significantly. So here's just a schematic of LCP, right? And so hopefully it provides this quasi-solid membrane environment with this viscous and sticky gel-like lipidic cubic phase. And so you see here that there are two non-intersecting networks of water channels, right? And so those are the sort of black circles shown in the lipidic cubic phase here. On the right, so let's see if I can use my cursor, you see this sort of egg-shaped thing going on, and so that's sort of like seeding going on in the lipidic cubic phase as a schematic. And so one of the cool things about this too is that because there are these networks of water channels, you can also get like feeding of more protein to nucleation sites with the lipidic cubic phase. In general, you'll get sort of better representative structures from using something like LCP to solve membrane protein structures, because the packing provides more hydrophobic contacts and lower solvent content, which is more like the native lipid bilayer environment from which they're extracted. And so it's better to use something like this where you're not using detergents to make the protein soluble. And so again, here's just another image showing different microstructures that lipidic cubic phase can assume. And so the one that's being displayed in this paper is the PN3M, so that's last one down here. So using lipidic cubic phase as a crystallization method of membrane proteins, you'll often get micrometer-sized crystals in initial screenings, but it will usually be very tedious and laborious to optimize those to be larger for use like at a synchrotron light source. And the resolution of these crystals is usually limited by the radiation damage that's inherent to all X-ray crystallography structure determination techniques. But again, because you get these micrometer-sized crystals in initial screening, if you're able to do that reproducibly, then those LCP-grown microcrystals are suited for serial femtosecond crystallography. So in serial femtosecond crystallography, right, it's this idea of diffract and destroy, where you're flying sample across the beam and pulsing, like the LCLS is pulsing in the protein and collecting diffraction patterns and random orientations of your protein molecule. And so your molecule sample is destroyed, but the diffracted photons have already exited the sample before they've been photoionized. The neat thing too about something like serial femtosecond crystallography is that the data collection is at a more native temperature, right? And so it's basically at about room temperature, whereas in traditional X-ray crystallography using like a synchrotron light source or even just a home source, you often use cryogenic temperatures where you're streaming like liquid nitrogen to prevent radiation damage. But here you wouldn't really care, right? Because you're destroying the sample as you're flying it across the beam. So you've extracted all the data that you need from that molecule. So sort of before this paper, SFX involved soluble proteins in aqueous delivery into the beam, and so that was mediated through this gas dynamic virtual nozzle. And so I'll talk about that in 2 more slides, but here's just an aerial shot of the XFEL, right? And so it was established by the Department of Energy at the SLAC National Laboratory in 2009. The peak brightness of it is 1 billion times higher than that of 3rd generation synchrotrons. And so you get the same amount of energy in 50 femtoseconds of XFEL pulsing that you would in like 1 second of synchrotron. It can pulse 100 times per second and deliver 10 to the 12 photons per pulse. And so it gives you this ability to diffract and destroy. And so here's just going back to the gas dynamic virtual nozzle usually used in aqueous soluble protein structure determination with serial femtosecond crystallography. And so I think Ganesh showed this previously in his journal club presentation. But it basically involves this gas focusing sheath, that supplies hydrated nanocrystals to the pulsed X-ray beam. Unfortunately, this isn't feasible for LCP because it's so viscous and sticky and gel-like. And so this brought about the need for an LCP injector, which is the focus of this journal article. And so here's the LCP injector setup. And so the injector here on the right is attached to a nozzle rod for insertion to the experimental chamber vacuum. What you're seeing in blue is a water line that provides pressure up to 300 psi, and it drives the hydraulic plunger that you're seeing here. You do have these 2 Teflon balls that provide a tight seal against LCP leakage. So this LCP reservoir in red can hold up to 200 microliters of LCP sample. The Teflon balls do morph though, so when you subject them to pressure, they form this like sort of cylindrical shape, and so those are changed out every time the LCP reservoir is replenished. And then in green here, the gas line that you're seeing is used for reliable extrusion of LCP. All right, so to make sure that there's this flow going, and then also to maintain coaxial flow, and so just to make sure that it's a focused stream, basically. And so here's just a video provided of this. And so I'll play it a couple times. But basically on the right, you're seeing the nozzle. And there's this green laser illumination that's been synchronized with the LCLS. And so this is sort of, you're seeing streaming going on of the human smoothened receptor protein that was used as a proof of principle for this journal article. And so this is flowing at 190 nanoliters per minute. And sort of 100 microns from or downstream from the nozzle, you see where the X-ray beam intersects the stream. And so whereas you have initially this continuous stream, you now see this like chopped up stream going on where the dark ripples on the jet here are a result of the X-ray pulsing your sample. So it's kind of hard to see that, but it's there. So the model protein that I've been talking about that they use in this journal article is the human Smoothened receptor. And so it's this GPCR-like frizzled class receptor. And again, it's involved in the hedgehog signaling pathway. And so it's really important for embryonic cell development and adult tissue maintenance. And so it has some implications in tumor growth because of this tumor maintenance signaling characteristic. And so cyclopamine is this ligand antagonist that was used in the structure determination proof of principle that's shown bound in the end result. And so, interestingly enough, this cyclopamine antagonist is actually used as a derivative for different cancer drugs, and there's plenty of literature on that. The cyclopamine ligand is naturally found in corn lily abundantly, and so you might remember a while back there was this one-eyed sheep, cyclops, basically, that resulted from its parent, like sheep, eating a bunch of this corn lily, and so it resulted in this developmental mutant phenotype where it's a cyclops now. And so these are just some images of the crystals grown for this paper. And so in A and B, you're seeing crystals that have been optimized for the XFEL studies, whereas in C and D, you're seeing crystals that were optimized for synchrotron use. And so the scale bars are 100 microns, if I remember correctly. And so some of the crystallization conditions are on the left. So they used a protein to lipid ratio of 2 to 3. And then the lipid they used is 9.0 mag with 10% glycerol. And also this was in 100 millimolar heaps, pH 7, 30% PEG 400, and 100 millimolar NaCl. So in the paper they mentioned that they did try to carry out they did try to collect data and determine the structure using synchrotron light source with the crystals shown here in C and D, but they were unable to do so. But obviously they went on to be able to do so with A and B using the XFEL light source. And so here's an image of some of the diffraction patterns collected initially from those microcrystals using the XFEL. And so it looks funky, right? And so the most commonly used lipid in LCP is this 9.9 mag. And so the problem with that is that it undergoes this phase transition into this LC crystalline phase at 18 degrees Celsius. And so that's an issue, right, because you're collecting data in an evacuated sample chamber at 10 to the negative 3 torr and at 20 degrees C. And so evaporative cooling can transform it into this crystalline phase. And you get this, as a result, you get these strong, sharp diffraction rings from that phase going on. And so it increases your background, and it poses a danger to the detector. So they did mention that they were able to attenuate the laser and collect diffraction up to like 2.5 angstroms, but instead they decided to do some things to sort of circumvent that phase change. And so they thought maybe if we change the co-flowing gas from helium, which is shown here in C, to nitrogen, then the formation of the LC phase is suppressed. And so it is. And what I took away from this is that on figure C here, you're seeing this birefringence going on. So those are the white specks that you're seeing, whereas you're not seeing it in D. And so birefringence, right, is crystals are characteristic of this birefringence. And so you don't see it any longer after you've switched out the gas to nitrogen from helium. But it didn't fully get rid of the LC phase. And so then they thought about replacing the 9.9 mag with a shorter chain mag. And so they used 7.9 mag 22 or 9.7 mag. I think ultimately they went with the 7.9 mag, which did end up causing the prevention of this LC phase transition altogether. And so then the diffraction patterns confirmed the expected cubic PN3M phase that I showed initially in those microstructures. And then the cool thing was that they discovered that you can add 7.9 mag after you've grown crystals and you don't observe any damage to your crystals upon addition. And so if, like, for whatever reason you can only grow crystals in LCP using the 9.9 mag, you can then go ahead and add the 7.9 mag and reliably collect diffraction patterns from the crystals that are not affected by this addition, essentially. So the LCP injector flow rate for the data collection was 170 nanometers per minute. It took from 5 to 10 hours for the data collection. And so the neat thing was that they used 100 microliters of sample which was less than 0.5 milligrams of protein, which is a vast improvement over the gas dynamic virtual nozzle requiring 10 milliliters to 10 mgs of pure protein to obtain a complete data set of your molecule of interest. And so these crystals were sub 5 micron, and they were collected at room temperature. So here are just some statistics on their results. And so there were 61,964 microcrystals used to recover the structure at this resolution. The resolution's not really high, and there are human smoothened receptor structures on the protein data bank at higher resolution. But again, this was a proof of principle trying to see, can we use this LCP injector in conjunction with serial femtosecond crystallography to reliably reconstitute the structure, to sort of obtain the structure. And so they solved it with molecular replacement. And from the 3,510,000 diffraction patterns, they ended up indexing 61,900 of them with this monoclinic lattice. And so the space group was P21. And so here's sort of the structure displayed as carton on the left. And so it's this gray carton on the left, and then the ligand is shown as green sticks. They did mention that the diffraction density of the ligand wasn't, I guess I could say, wasn't good enough to where they could determine the orientation of it. And so they had to sort of think about it, thinking about sort of more like the geometry of the ligand. And the one thing that I was going to mention, I guess, about the ligand binding here in this figure on the right, figure B, is that it might have been nice to have shown the polar contacts here that are being shown in gray. I'm just trying to outline them with the cursor as black as opposed to gray, because it sort of makes it more difficult to see. And so I think the paper is fantastic, and I'm not going to offer criticisms on it, but this is one thing that I guess I would have done differently. It would have been much more clear to see if they were just black, especially when you print off the paper. And so the implications of this paper, right, is that previously SFX was possible with aqueous soluble proteins delivered to the XFEL using this gas dynamic virtual nozzle. But now with this proof of principle study, they're able to do LCP SFX studies. And so you get this dramatic reduction of protein sample required. And sometimes it can be difficult to obtain large amounts of pure protein for crystallization, right, for various reasons. Sometimes it doesn't purify well just because, or it doesn't express well, just because like the protein you're expressing might be toxic to the cell line you're using. So sometimes E. coli doesn't tolerate certain protein overexpression. Or maybe your protein might require certain like post-translational modifications if it's like a eukaryotic protein. And so you sort of lack the ability of a robust cell line that can make a lot of this protein. And so as a result, you can expect an increase in the number of structures that are determined using LCP and SFX, especially some that were difficult to optimize for larger crystals and use at a synchrotron light source. And so that's all I have, and so I'm willing to take any questions.